All right, hey everybody, welcome. Tonight we're going to talk about berry development and the ripening process for grapevines. And this, of course, is for our Vidi 303 class covering everything that happens in the vineyard from harvest to dormancy. So, outline of what we'll talk about tonight, we'll talk about grape cluster and flower anatomy, berry formation, which includes anthesis, pollination, fertilization, and fruit set. We'll talk about the 3 stages of the development of the berry once it's formed and how it ripens, how to determine ripeness of the fruit, and then tracking grape sample data. So the cycle of the grape. Most commercial grapevines are examples of what we call perfect flowers, and that's because they contain both male and female organs, so they are hermaphrodites. What makes them a perfect flower is that they are self-pollinating, so they're extremely efficient. It is actually believed that insects are not necessary for pollination, just like a gentle breeze and good spring conditions will do the trick, which is pretty cool. Going on to grapevine cluster anatomy. Here we have like the shoot, which is like the main axis which the leaves and grape clusters are set on. Then we have the peduncle, which is the stalk that bears the fruit. So it's just this little section here. Then we have the rachis, which is kind of the core of the body of the cluster. So it's the main axis. This accounts for 2 to 5% of the fresh cluster weight. Then we have what's called the shoulders, which are the prominent branches arising from the rachis, typically the top section here. Then we have the pedicel, which is where individual flowers attach. And then we have the flower itself, which will self-pollinate and then eventually become a delicious grape. So this is a figure from a typical Thompson seedless flower cluster before anthesis. So flower anatomy. Here we have a flower, very typical, perfect flower for grapes. And it contains both male and female parts. Of course, we have the stamen, which is this large section here, which includes the filament and the anther. The filament supports the anther. The anther contains the pollen needed to fertilize the female parts, also known as the stigma. This receives the pollen. The pollen travels down the style, which it fertilizes the ovary, and then it becomes an embryo sac with a single egg and eventually becomes fruit. Interestingly enough, the grape flower contains odor glands that produce no nectar, which is interesting. It's called the nectary. So many Vitis species secrete odorous compounds to attract insects. Vitis vinifera seem to have no response. So other grapevines, besides the ones that we use for winemaking, do produce compounds to attract insects, but for whatever reason, Vitis vinifera just doesn't really seem to bother with that. Next we have the pedicel, which is the attachment for the flower, right? Very cool. So now that we know a little bit about the anatomy of these things, let's talk about like the process of the flowering, the opening, and the fertilization of that. So the process or the act of opening a flower bud, once it's ready to become fertilized, right, the protective cover will come off and expose a beautiful flower. This is called anthesis. What's happening is there is a cap protecting the flower called calyptra, and it contains 5 fused petals that protect the flower organs until it's ready to open up in the spring, and then they'll kind of curl up like a cute little hat, and then it will eventually pop off and reveal the opening of the flower bud. This typically happens around 6 to 8 weeks after the beginning of shoot growth. After the flower is open, the anthers will release pollen that's received by the stigma. Pollen germinates on the stigma and creates a pollen tube. It works through the stigma and style, ultimately reaching the ovule, so 2 to 3 days. Then fertilization occurs and the berry begins to form. There is like an optimal temperature and like weather conditions for this. 80 to 90 degrees are typically best according to the New Viticulture textbook. So, interestingly enough. Fruit set. This is a stage in the grape development process. It's like when the flowers fall off and you start to have fruit show. And it looks very nice, like this left picture, right? So this is a fully fertilized cluster. So every single flower was able to fertilize and produce a fruit. and it looks like these little green harsh berries. Do not eat these. These are going to be extremely rough. They're definitely no sugar yet, right? Here on the right, we have a cluster with lots of shatter. This is what happens when flowers fail to fertilize and they just fall off. So it's called a shatter there. This could be due to the climate, rain, hail can cause this, right? If the temperatures go in either extreme, too cold or too hot, it can also fail to fertilize, so it's not great. Typically, 20 to 30% of flowers will develop into berries. Cool. Fruit set is also defined as the stage when berry diameter is between 1.6 and 3.2. Not that we would necessarily be out there measuring all those individual berries. You just know from looking at the cluster, it looks like this, right? You have berries set, you got fruit set. Awesome. There are other situations where you can find problems with pollination in a vineyard, and this is a very common term called hens and chicks. This describes the uneven pollination that can happen in vineyards in unideal spring conditions, right? So again, bad weather, nutritional deficiencies, particularly boron is one of the leading deficiencies, viral infections. In French, it's called melandage, which is the same thing, right? Definitions, this would be bunches that contain berries that differ greatly in size and maturity, and then small seedless berries amid normal berries. So these would be berries that failed to fertilize. Okay, as far as the anatomy of the grape itself, the fleshy berry, as it develops, it goes through three main phases, which we'll talk about in the next slide. But as far as the anatomy goes, there is the epidermis, which is a lot like the anatomy we talk about with humans, right? It's the outer skin covering, or the cuticle covering in this case. Then we have the hypodermis, which is just under the epidermis, comprising several cell layers. Then we have the mesocarp, which contains larger vacuoles and comprising of 85 to 87% of the berry's spherical volume. The endocarp, which consists of crystal containing cells, juices, and an inner epidermis of itself. And then we also have seeds, right, contained in locules and composed of an outer seed coat, the endosperm, and the embryo, right? So ultimately, what I want you to take away from this is, there's an outer skin. There's a couple of different layers inside the berry. We have seeds. These seeds have a coating that make them indigestible, right? The pedicel, this is where it attaches to the grape. And then it's on the direct other side of this pedicel, we actually have something called the style or remnant scar. And that is actually a scar left when the calyptra, that cap of the flower comes off. That's a scar from that, which is pretty interesting. Okay, what is a really important takeaway is this berry ripening curve. So grape berries ripen in 3 stages, right? We have a couple of things going on here. So here on the vertical axis, we have berry size and coloration, right? So it starts at really small and green, and then it becomes really large and purple in color, right? We have this map or this curve set into 3 stages here, stage 1, stage 2, stage 3. The blue line represents the concentration of organic acids. The red line represents the concentration of sugars in development. And then veraison is highlighted because it's the stage where the berries start to change color, right? So that's kind of what's going on. All of these compounds on the bottom, not so much important for you to memorize for this class. All this means is these are the compounds that are developing in this specific stage of the berry development. So stage 1, we see, very little growth, and we see acids start to rise, right? So acids are rising, acids peak in stage 2. We haven't had any sugar yet. Stage 2 kind of ends when we see veraison, so when the color of the grape starts to change. And then in stage 3, we see acids plummet and sugars start to rise. So those are the general curve of grapevine production, berry development. So specifically explained, right, stage 1 is berry formation to lag phase. So cells begin to divide in the berry, acids and tannins develop in the grape skin and pulp. Stage 2 is lag phase to veraison. Berries will reach half of their final size, cell division will stop, but they'll start to enlarge. So they start to actually take on a lot of water, they grow. Seed embryos begin to form and grow. Cells accumulate more acid and tannin, reaching peak content before veraison, right? So they're adding water, more acid, more tannin. The seed embryos are growing. And then at stage 3, we call this post-veraison ripening. So this is marked at veraison, so that's right where stage 3 starts, when berries change colors, soften, accumulate sugars, and they start to metabolize acids. This is where the inverse relationship of sugar and acid comes into play, right? And then it isn't until some of these later stages that we see something called engustment, which is where the berry skins start to develop aromatic compounds or flavor compounds, so you can see. Okay, so why is this important, right? Because we're monitoring the levels of sugar and acid in a grape to determine when it is the optimal time to harvest, but also it's a chance to see like optimal conditions for grapevine production too, right? So here on this top graph, we have representation of optimal ripening of a grape. And that is, as a grape ripens, sugars raise and acids start to diminish. When it comes time to pick, the sugar to acid ratio will provide an ideal ripeness, flavor, and color to the grapes, resulting in high quality wines. So it's being picked at a time where sugar is ideal, acid's ideal, and there's plenty of flavor in the skins, and that is due to good viticulture practices, a good climate for producing, right? It's just a perfect combination of everything coming together just as we need it to, right? Here on the lower graph, we see the same grape under warmer conditions, right? So if the conditions are non-ideal, the weather is too hot in this situation, the sugar content will be out of balance with acidity, leading to unbalanced fruit. It can also happen if the climate is too cold too, right? But this situation is showing you too hot, for example. So in extreme warmer conditions, the sugar will be too high, acids will be too low, and the flavor of the grape will yield dried fruit or prune-like flavors, right? So it's unideal. So here we have the ideal sugar to acid ratio is right here, but unfortunately, an ideal flavor was here. However, grape develops extremely quickly, fails to reach ideal color, and then shrivels. So, not a great time. Okay, we talked about this in this class already, vine balance, but it is important to talk about for grape ripening, because if you overcrop, you're not going to have a lot of flavor in your resulting wine. That's the big thing I want you to understand. It's the grapevine's taking on a load that it can't manage, it can't ripen, because its first priority is to reproduce. So it's producing as much fruit as possible to spread its seed and, get these plants out in the world, but it can't make good wine that way, right? So some grapevines are worse about this than others, like Zinfandel is an overproducer for sure. So that's when we'll have to drop crop. And we typically do this at veraison. So right when the skins start to change color, we'll drop them and in hope that the rest can ripen correctly. Cool. When to pick. So you'll want to keep a data log of samples that you take from the vineyard. You're going to want to track your sugar level, pH, and also seed maturity. You can try your best to kind of forecast or predict when you can pick your grapes based off of the previous data, and you can see like how much sugar you're accumulating every week or every day, and you can go from there. So berry sampling is very important. We did talk about this in lab for our Bricks lab in person, and also for pH, right? So it's really important to take measurements of the progress of your fruit. This will help determine when to harvest. At this point, for this process, you would go out into the vineyard with a Ziploc bag and then pick random berries to obtain the most accurate sample for analysis. Some other forms of sampling include taking the entire clusters, picking all the clusters off of a single vine, right? Every vine and location will have variation in sugar, acid, and pH, right? And that makes sense because every single berry was fertilized at a slightly different time. So depending on when that berry was fertilized, just like this picture up here, it might actually be in a different stage of development than a couple berries over from it, right? So this is like kind of in a bizarre situation. This is a veraison, right? But some of these berries fertilized way before the others. That's why you're seeing that. Okay, so compositional factors that we look for, we did talk about this in class, but we'll cover it again. Sugar, which is Brix content, pH, seed maturity, and taste test. We're also going to do a titratable acidity lab in this class because that is a component that buyers of grapes would want to know before they harvest as well. Okay, so Brix. It's important to know the Brix content. Obviously, 1, because it determines the ripeness and maturity of the grape. 2, because it will help indicate what style of wine you're going to make and the flavors that will come with it. So whether you're making sparkling, a late harvest, a white, rosé, or a red from a specific grape variety, those will all be picked at different times at different Brix contents. So for example, a sparkling wine is picked very, very early. It's low alcohol, very high acid, which means low Brix, high acid. Late harvest, as the name implies, is picked very, very late. High sugar, lower acid, and also like the grapes could be shriveled on purpose too, right? So depending on what style you want to make, it's important to know these numbers. It's also the best way to determine the final alcohol concentration in our wines, which we discovered with the formula potential alcohol equals initial Brix times 0.6. Oftentimes, Brix is measured with the pocket refractometer. Of course, this is best for juice samples only, not fermenting musts. And then we also have a hydrometer, which is shown in this photo, and that is commonly used for fermenting musts. Okay, next we have the pH. pH, in general, if you've taken other science courses or just have an understanding of some basic science, reflects the chemical conditions of a solution. It's put onto a logarithmic scale ranging from 1, which is highly acidic, to 14, which is highly basic, with 7 being neutral. The reason that this is important is because this is a major indication of health for the juice and the resulting wine. We want the pH to be in a certain range for our fermenting must to be healthy, for the yeast and bacteria that we're using to be happy and be promoted, right? So all these functions are reliant on the pH of the solution that they're living in, right? Also, lower pH helps preserve wines during aging and keeps it from spoiling, right? And that's because it's in a state where, spoilage yeast or bacteria cannot thrive in. That's why it's important. Okay, here we have like a pH scale just in case you needed a little refresher. Anything below 7 is acidic. Anything above 7 is basic. Wonderful. Next we have seed maturity. It's important to look at the seeds because you get some tannins from the seeds as well, especially if you're making a red wine. The seeds can indicate when the grape is mature and it was intended to be eaten, I guess, right? So tannins come from the seeds, but also the skins of the berries. These are a mouthfeel sensation. It's a texture component in wine. So if you had picked grapes where the seeds were still green, they were not ripe, you would get some really harsh tannins in your wines, right? The seeds are brown, they're more mature, and it'll be better integrated into your wine and be more pleasant tasting, right? Better mouthfeel. Also, something to note too, the pulp of the grape will come off of the seed very easily when the grape is physiologically ripe, when it's determined, right? Cool. Some people pick by taste, which is very fun too, and there is merit to this. So one of the final stages of berry development is engustment, which we talked about earlier, right? This is the stage where aroma and phenolic compounds start to accumulate in the berry. So both the aroma compounds and then like color and tannin. This happens before berries start to raisin and develop overripe flavors. And if you're very experienced or just very confident, grape growers will choose to pick off of the taste as well, or just purely alone on the taste. All right, next we just have our learning objectives from this presentation. Just one slide of objectives to go over before you take the quiz. And then I also have some resources. I referred to the New Viticulture book by Jamie Good, Chapter 7, page 111. A lot of great resources. It's a very old textbook, but it still has a lot of great information. And then we also have the UC Davis Grapeberry Growth and Development PDF linked here if you'd like to learn more. Also a great resource. So I hope that was helpful. If you have any questions, feel free to email me. But otherwise, I look forward to seeing you guys next time.